Hello, folks, and welcome to part 3 of our unit on gene expression regulation. And this one, we're going to do part 3A, gene expression. We're looking specifically at the regulation of genes and when they're expressed. Most of the stuff you can get in the beginning of chapter 18 in the book. In part 3B, we'll go over 6.6, gene expression, and how that influences cell specialization. That'll be a little bit shorter section. And that's from chapter 8.4 in your book. Let's begin. Can we control when genes are expressed? Yes, we can. Prokaryotes and eukaryotes precisely regulate their gene expression in response to environmental conditions. So we can determine when to turn genes on and off, not consciously, obviously, but in response to environmental conditions. That doesn't just mean like, out here environment, it means in here environment, the environment inside your cells and inside your body. In multicellular eukaryotes, like us, gene expression also regulates development and is responsible for differences in cell types. I mean, we've got skin cells, liver cells, muscle cells, nerve cells, and those all have all the genes, all the DNA that every other cell has, but we only express the genes that help it work as a nerve cell or a muscle cell, for instance. RNA molecules play many roles in regulating gene expression in eukaryotes, and we'll learn some of those here in this section. Prokaryotes often respond to the environmental change by regulating their transcription of the proteins, so whether or not they transcribe them or not. Natural selection has favored bacteria that produce only the products needed by that cell. Why waste energy on materials you don't need? So if you made everything all the time and you didn't need it all the time, that was a waste of energy and you were less likely to survive. So those who didn't waste that energy on stuff they don't need lived. A cell can regulate the production of enzymes by feedback inhibition and gene regulation. So we'll get into that in this section. One mechanism for control of gene expression in bacteria is something we call the operon. So let's take a look at the operon model. Well, actually, before we get to operons, let's take a look at kind of some rapid and long-term response. So here we've got our precursor molecules that we're going to change into tryptophan down here. There are 3 enzymes involved in this. So first you take precursor enzyme 1, makes it look like that. Enzyme 2 makes it look like that. Enzyme 3 does the final bit. These are the genes here. See, they're on DNA. The code for making enzyme 1. This one codes for making the second enzyme. This one codes for making the third. Okay. So rapid response. If we need to quickly shut down, I know it's shut down because I got a negative here. If we need to quickly shut down the production of tryptophan, if we have too much tryptophan, it will, just the sheer presence of a lot of tryptophan will block the function of enzyme 1. Now, that'll take care of it quickly, but it's not a long-term solution. The second tryptophan goes back down, this will start going up again. The other thing is a lot of tryptophan will actually start to reduce the transcription of these genes, making these enzymes, thus reducing the amount of tryptophan being made. So this is quick response. This is long-term response. So let's talk about operons. They are a cluster of functionally related genes that can be coordinated by a single on-off switch. So we can turn on the switch and all these related genes that work on, helping you digest sugars get turned on, or we can turn it off and none of them get expressed. Okay, that switch is something we call an operator. Usually has a promoter there, something that promotes it being used. An operon is the entire stretch of DNA that includes the operator, the switch, the promoter, and then all the genes that operon and promoter control or turn on and off. So you say on, transcribe these buggers. Off, you don't transcribe them. We can switch it off by a protein called a repressor. It represses the operon. The repressor prevents gene transcription by binding to the operator and blocking RNA polymerase. So physically, it gets onto the operator, and when it's on there, the RNA polymerase can't lock on to try to make RNA. It's just like, something's in the way, I can't get there. So it's pretty easy. The repressor is the product of a separate regulatory gene. So you have another gene that makes the repressor, which can then repress that one. So there's a couple of steps in there. Your repressor can be active or inactive, depending on the presence of other molecules. There's also things called co-repressors. which is a molecule that cooperates with the repressor protein to switch an operon off. So, in other words, it's not as simple as just a single molecule doing a single thing. Okay? We can have, sometimes in order for the operon to be turned on or off, we need other molecules there. Sometimes they're just the molecules that it's keying off of, and other times you actually need it. It actually helps repress. By default, the tryptophan operon is on. Okay, so it's just left on, and the genes for tryptophan synthesis are transcribed, and you're making tryptophan. Okay, so when tryptophan is present, it'll bind to the repressor, which will turn the operon off. So if you've got a lot of tryptophan, it'll bind to the repressor protein, which will then grab onto the operon and turn it off. So unless you have enough tryptophan to do that, you're just going to be making tryptophan. It's not bound, the operon's on, and it goes. So you get a lot of tryptophan, binds to the repressor, which grabs on and blocks it so you can turn it off. Okay. The repressor is active only in the presence of its co-repressor, tryptophan. Both of them need to be there. Thus, the tryptophan operon is turned off or repressed if tryptophan levels are high. So we can see that here. Here's our promoter. Here's our regulatory gene. Okay. Here's the operator, an RNA polymerase. So normally, the polymerase can grab on and it can go down and transcribe all of the mRNA we need to make all of these different enzymes. Okay. Here's the inactivated repressor right there. It's not doing anything in this situation because we don't have any tryptophan to activate it, to activate the repressor. Okay. Down here, we can see now we've got tryptophan present. Tryptophan can now bind to the repressor and activate it. Now it changes the shape of this. You see how it was curved? Now it's straight. Now it can fit into this spot on the operator, right there. And that physically gets in the way of the RNA polymerase getting in here to keep going down and transcribing this mRNA to make these proteins. So this is the default position. No tryptophan, but as long as there's enough tryptophan, it can bind here and block there. So repressible and inducible operons, okay, which are two types of negative gene regulation. So, you know, if you're getting too much of something, it'll make you make less, or if you're getting too little of it, it'll make you make more. That means negative, like negative feedback loops, which you mentioned earlier in the year. So a repressible operon is one that is usually on, like we just talked about. Okay, the binding of a repressor to the operator shuts off transcription. So the repressor represses those operons, turns things off. An inducible operon is one that's usually off. Okay, so we have a molecule called an inducer, okay, that inactivates the repressor and turns on transcription. So in that one, the repressor is usually stuck on there and blocking transcription, so it's stuck in the off position. And in order to open it up, you got to get that repressor off. And what you do is you got to, the inducer will come in and basically pry off the repressor, so the operon is free and can be transcribed. Okay. Something we call the Lac Operon is an inducible operon, like we just talked about. It has genes that code for enzymes used in the hydrolysis and metabolism of lactose. So breaking down lactose, the sugar lactose in milk. Okay. By itself, the Lac repressor is active and switches the Lac operon off. So here's our DNA. Here's the operator right here, and the repressor is stuck on there. It's grabbed on and it's blocking it, and we can't do anything. Okay, so the inducer will pry this repressor off, and then that gene can be transcribed, turning on the Lac operon. So here we got the active repressor. When nothing is bound here, it sticks to the operator and blocks it. If allolactose, so we got lactose present. So normally we don't have lactose, don't need to worry about digesting it. But when lactose is present, a molecule of allolactose can bind to there, change the shape of the repressor, which disconnects it, and then the polymerase can go ahead and make everything in that particular operon, these three enzymes, which is pretty cool. Same basic function, it's just whether they're normally on or normally off. Okay, and there's either inducer or repressor here. Inducible enzymes, like we just talked about, this one here, which makes these things, which help to break down lactose, are usually functioning catabolic pathways. Remember, catabolic pathways are pathways that do breakdown functions, like breaking down a big lactose molecule into smaller molecules that are more digestible. Okay. Repressible enzymes usually function in anabolic pathways, or ones that build things up. Okay. Synthesis is repressed by high levels of the end product. So if you get too much of the end product, it'll shut them off. Okay. Here, when we want to break things down, it's usually that thing that we want to break down that's going to send the chemical signal, hey, we got this stuff, so let's induce that thing to break it down. Here, we're constantly making stuff unless we have too much, and then we shut it off. So those are both negative control mechanisms. Positive gene regulation, or positive feedback, through a stimulatory protein, like CAP, an activator of transcription. It starts transcription process. So when glucose, the food source of E. coli, what it prefers, is scarce, doesn't have enough sugar to eat, CAP is activated with cyclic AMP. Okay. That activated CAP then attaches to the promoter of the Lac Operon and increases the affinity of RNA polymerase, thus accelerating transcription. So it doesn't just turn it on. Okay. It literally makes it more active. So the other ones here is just turning it on or off. This activator actually gets this gene to be more attractive to RNA polymerase. So you're going to make more RNA than you normally would. So it increases it because, hey, we don't have enough sugar. We've got to do whatever we can to break down any lactose that's around. So here you can see the activator, active, bang, helps to bring this polymerase in to bind it. All right, guys, let's take a look at gene expression and specialization in eukaryotes. And we can regulate gene expression at many different stages. There's possibilities of getting in there and determining whether we get from the DNA signal code all the way to that finished final protein or product. All right. In multicellular organisms, regulation of gene expression is essential for cell specialization. If you want to have a liver cell, you better only express liver genes and not nerve genes, for instance. Okay. Almost all cells in an organism are genetically identical, okay, having the same DNA, all 46 chromosomes. Differences between cell types results from differential gene expression. We express liver genes in livers and nerve genes in nerves. Okay. Abnormalities in gene expression can lead to diseases, including cancer, when you're replicating faster you need to, right? And gene expression is regulated at many stages. So here we can see some of the different stages going on in this whole process. And at any one of these, we can get in and change something that changes whether the gene gets expressed or not, or gets modified. So you got signals, you got the DNA, you got transcription, processing. Then we're going to transport it out to the cytoplasm. Once we're in the cytoplasm, we go through translation. Do we break the RNA down right away, or do we let it sit around for a while? We make the protein. We activate the protein. We change its shape so it can do something. Do we let it keep doing that, or do we break it down? All sorts of different places in here where we can get into the middle of this cascade of events and change things. And let's take a look at some of them. Okay, right off the bat, we can regulate chromosome structure. So chromosomes, long strands of DNA, right, are twisted and wrapped around themselves really, really tight most of the time. Okay, which makes it difficult for transcription to happen. Okay, so in order for transcription to happen, you have to unwind it so you can split it and expose those things and actually transcribe some mRNA out of it. So genes that have highly packed heterochromatin are usually not expressed. They're too dense. It's hard to get at those genes. We can chemically modify those histones, which are the proteins that help hold that DNA in that tight helix. Will alter both the chromatin structure, how tight or loose it is, and thus affect gene expression. Okay. So you can loosen chromatin structure, which will promote the initiation of transcription or make it easier for transcription to start to make that mRNA. If you add methyl groups to DNA, it can condense the chromatin, which has the effect of reducing or blocking transcription because you can't get it opened up to get at it. If we phosphorylate next to the methylated amino acid, we can loosen the chromatin, and then we can transcribe it more. And here we can just see these histones wrapping up the DNA. So this section would be hard to transcribe. This section would be easier to transcribe. Like I said, methylation reduces transcription. And we can use that for long-term inactivation of genes and cellular differentiation, like we just methylate the genes that deal with making nerve cells in our liver cells. Something else interesting there is we can actually do genomic imprinting, where methylation regulates expression of either maternal or paternal alleles of certain genes at the start of development. So in some species, in some genes, we can say, oh, we're going to go with the mom's genes, or no, we're going to go with the dad's genes. Those are the ones we're going to transcribe, not those ones. This leads to something we think of or call epigenetic inheritance. Although these chromatin modifications do not alter DNA sequence, they don't change your DNA at all. Those condensations or relaxations can be passed on to future generations of cells. That inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence, we call epigenetics, okay? Which is very interesting. I don't know a lot about this subject. This is one that I think is very fascinating. I'd like to do more research on. If you want to do more research on it, that'd be great. But we think this might have something to do with why you can have generational trauma. Okay, part of it is nurture, gets passed on in behaviors. Part of it might be trauma and exposure to certain things can cause changes to this type of stuff in your DNA. Not directly mutating the DNA, but changing where it's tight and where it's loose and those kind of things, which can then have effects on your kids and your grandkids. A few generations out, eventually it disappears, but there does seem to be a correlation. I don't know a ton about that one, though. That's one we should look into. All right, so that's what we just said. Chromatin modifying enzymes provide the initial control of gene expression, either exposing the DNA so you can transcribe it, or not exposing it so you can't. Okay, once we've done that, how do we organize a typical eukaryotic gene? Has a lot to do with whether it's expressed or not, or how it's expressed. So associated with most eukaryotic genes, we have control elements. Okay, we talked about operons in bacteria, right? These are kind of the same thing, but a bit different. They're segments of non-coding DNA, they don't code for a particular protein, that serve as binding sites, a place for the transcription factors that are going to help transcribe things to attach to the DNA and then run down it. Okay, they're the docking port. Control elements and those transcription factors are critical to the precise regulation of gene expression in different cell types. Just going to show you here, there's here's our exons and introns. Here, this is our what's going to be transcribed. There's the promoter that starts it, and we have some control elements right next to it. And we also have some that can be far away up here, which we'll look at in a little bit. So to initiate transcription, eukaryotic RNA polymerase requires the assistance of transcription factors. It can't just come in on its own and grab on and start transcribing. You need to have some help. And that help is transcription factors, which help it all to get into place and do its thing. General transcription factors are essential for the transcription of all protein coding genes, but we also have particular ones that have specific control elements interacting with specific transcription factors for genes that we want even more control over. Proximal control centers and elements right here near the promoter. Remember, that's kind of like we're going to start transcribing. That's what the promoter does. Distal elements, which we call enhancers, may be far away from a gene and even located in the intron. Okay, may not code for anything. So an activator protein, which I have pictured down here, has two parts. One domain binds to the correct part of the DNA. So it says, oh yeah, this is the promoter region. This is where we need to bind to. And then they have the activator domain, okay, which actually help to bring in those transcription factors, okay, and result in the transcription of a given gene. So let's watch a short video on it, and I'll meet you on the other side.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
So here we can see that we got our gene, our promoter, which includes a Tata box. Okay. And then upstream of there, we have our enhancer. So activators come in. They help bring in general transcription factors, bind it all together, and help the RNA polymerase get all bound up where it needs to be so it can start doing synthesis or transcription. Some transcription factors function as repressors, and they can actually inhibit the expression of a particular gene, blocking it basically. Some activators and repressors act indirectly by influencing chromatin structure to promote or silence transcription. So all these different factors can bind up and either help get transcription going or bind up and stop it, block it. Okay. But the same basic mechanisms of bending and having the right shapes to fit onto certain things, either to fit on to block things or to fit on to help other things fit on. Okay, give them a hand to grab onto the rope, so to speak. Combinatorial control of gene activation. Okay. A particular combination of control elements can activate transcription only when the appropriate activator proteins are present. What the heck does that mean? Well, let's take a look at this picture. So here we are looking at a liver cell over here and a lens cell over here in the eye. Okay. So in the liver cell, we've got the same gene, or we've got here enhancer for albumin gene, enhancer for crystalline gene right here, the promoters, and here's the genes. Both cells have both these genes and both these enhancers and control elements, right? We got the same DNA strands in all of our cells. So they both have all of this. But in a liver cell, only that liver albumin one is expressed because it has the right activators to allow it to work and hook up. Lens cell, or in the liver cell, we don't have the activators we need to turn on the crystalline gene. The opposite is true in the lens cell. The available activators up here, they won't interact with this, so you're never going to turn this gene on, or transcribe it, I should say, but you will turn on the crystalline gene. So it's a big part of specialization. Coordinatedly controlled genes in eukaryotes. So this is kind of like in the bacteria, on the prokaryotes, we had an operon, which was the operator, right? The on-off switch, and then it had a whole series of related genes after it. So like that tryptophan, we had all the genes for like the 4 different enzymes that need to be produced to make tryptophan. Were all together. You turn that on and you transcribe all 4 of them at once. In eukaryotes, we're not organized like that. There's some exceptions, but we're not really organized like that. These genes that need to work together to get something to work can be scattered over they can be together, they can be on the same chromosome, but in very different places on the chromosome. They can even be on different chromosomes. Okay, but the reason they all express at the same time is because they have the same combination of control elements. So if you got the right mix to turn on the control element for that gene, it's also going to be the right mix of stuff to turn on the transcription of the other 4 genes you need to make this, hemoglobin molecule, for instance, in blood cells. Okay. So copies of those activators recognize specific control elements and promote simultaneous transcription of the genes. So you got the same key, so to speak. So if you've got that key, you can fit it in these 4 different locks on different places and open all those doors or turn on all those genes. Okay, so that is regulating whether we turn these genes on or off. So now let's assume, and that's where we do most of our regulation, but we can also regulate after transcription. So post-transcriptional regulation. Transcription alone does not account for full gene expression. We can have many regulatory mechanisms that operate at various stages. And they allow you to kind of fine-tune gene expression. And this is more common in rapid response to environmental change. If you remember on the very first picture slide we had in this PowerPoint, I had the tryptophan, right? There was the fast response, and then there was the slow response. Slow response was altering DNA expression, making those enzymes. The fast response actually blocked one of those enzymes and inactivated, so you couldn't complete the process. And that's what we're talking about here. One of them is alternative Rna splicing. So different mRNA molecules are produced from the same primary transcript, depending on which RNA segments are treated as exons and which as interons. So here we've got our 1, 2, and 5 are exons, and there's 3 and 4. Okay, that's our DNA. Here's our primary transcript. We have our interons here they're not going to be used, but 3 and 4. In this one, we've got the 3. In this one, we have the 4. So these 2 mRNAs are going to wind up coding for different proteins or polypeptides, even though they both came from the same primary transcript and they both came from the same DNA. So we're getting it. So this transcript has already been transcribed, but with this RNA processing, we can alter it a bit. So here's a video. I'll catch you on the other side.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, a cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript.
So, some other things that we can do after transcription. We can affect the initiation of translation, and we can affect how long the mRNA sticks around or when it starts to degrade or fall apart. So the initiation of translation of selected mRNAs can be blocked by regulatory proteins that bind to sequences or structures on the mRNA. So we can, once again, just get a protein grab onto this mRNA and kind of block it from being able to do what it wants to do. It's holding your hands like a pair of handcuffs, right? Doesn't let you move where you need to move. Alternatively, translation of all mRNAs in a cell may be regulated simultaneously, like at the initial activation following the fertilization of an egg. We just all go crazy. They all get turned on. Okay. The lifespan of mRNA molecules in the cytoplasm is also a key to determining protein synthesis. Eukaryotic mRNA, by the way, is more long-lived than prokaryotic mRNA. Eukaryotic cells are much more longer-lived in general. Nucleotide sequences that influence. Let me back up. So first off, if your mRNA only sticks around in the cytoplasm for a little bit of time, each mRNA molecule is only going to be able to cause the synthesis of a few protein or polypeptide chains. But if that mRNA molecules can last for a day or 2 or whatever, it's going to be able to make polypeptide after polypeptide after polypeptide after polypeptide over and over again, and you're going to get a lot more of that protein. So it's not necessarily that we turn that gene on or off, but at this stage, we can affect how potent that gene was. If the mRNA has a shorter lifespan, less potent. It's not going to make as much protein. It's got a longer lifespan. That gene winds up being much more potent. Nucleotide sequences that influence the lifespan of mRNA in eukaryotes reside in the untranslated UTR region at the 3' end of the molecule. So we can watch this video here and you get a little bit of that.
The lifespan of mRNA molecules helps determine how much of a protein is made. Nucleotide sequences that affect mRNA longevity are often found in the 3' untranslated region following the stop codon. Enzymes attack the poly A tail first and eventually the rest of the mRNA. Depending on its ability to resist this degradation, a eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made.
All right, so now we can go to the after translation part. Okay, we all know that protein, once it comes out, is not complete. It's got to fold. It's got to maybe sometimes bind with other proteins and things like that, and get processed before it can do its thing. So we can affect regulation of that gene, or how much influence that gene has on the final protein product, by regulating the degradation of that protein, just like we did with mRNA. We can get rid of it. This mark that tells you to get rid of it is recognized by proteasomes. Don't think you need to know that term, which recognize and degrade the proteins. Just got a video here that kind of shows what that looks like, and then we'll see you on the other side.
A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
Briefly want to talk about something called non-coding RNA. Non-coding RNAs play multiple roles in controlling gene expression. In reality, only a small fraction of our DNA codes for full proteins, and a very small fraction of the non-protein coding DNA has genes for RNA, such as rRNA and tRNA. A significant amount of the genome may actually be transcribed into something we call non-coding or ncRNAs. Okay? These non-coding RNAs, we think, play a role in regulating gene expression at 2 points: mRNA translation and chromatin configurations, or that tightening and loosening of the chromatin we talked about before. So they're just one of the factors that are involved in that. But let's talk about mRNA translation specifically here. These microRNAs, or non-coding RNAs, these really small ones, are small single-stranded, flipping my words again, are small single-stranded RNA molecules that can bind to mRNA. They can do one of two things. They can either degrade the mRNA, break it apart. We talked about before, a shorter lifespan for that will mean it doesn't produce as much protein, or it can block its translation. It can glom onto it and keep it from being able to bind up in a ribosome to translate into a protein. And there's some estimates that put expression of at least half of all human genes may be regulated at some level by microRNAs. And here we can just see some images of that. So here's our microRNA along with the protein that it attaches to. It can grab on to our mRNA and break it down, or it can grab on and keep it from being able to get into the ribosome. So just some implications of all this stuff we've been talking about, guys. So just a couple things. The phenotype of a cell or an organism, so what it looks like, how it behaves, what it does, is determined by the combination of the genes that are expressed. Some are expressed, some aren't, and the levels at which they're expressed. So how much they're expressed. Are they expressed just a little bit or expressed a lot, make a lot of protein from that one gene? Observable cell differentiation, so noticing the differences between different types of cells, results from the expression of genes for tissue-specific proteins. So that's how we get specialization. Okay? Liver cell, lens cell, nerve cell. Induction of transcription factors during development results in sequential gene expression. So this gene will express, which will cause this gene to express, which will cause this gene to express. And that plays a role in, well, I don't want to get too far, during development. So without going too far into that, I want you guys just to either, if you're on your own, stop for a bit and think about the implications and importance of these above statements. Or if we're in a class, you can get together and discuss it with some classmates. What are some of these implications for development over the lifespan of organisms, for evolution, and things of that nature?